my name is Bill Montalbano, and today I'm going to be talking about enveloped viruses versus non-enveloped viruses.
Alright, so I'm going to start out by talking about the virion. The virion is just a single virus particle. It consists of a viral genome, which can be either DNA or RNA. It is inside of a structure called a protein capsid. So this whole light gray area surrounding this darker black area is called the capsid. The part on the inside is called the viral genome. The capsid just provides extra protection for when it enters into a host cell. Viruses that only have the protein capsid for protection are considered naked or non-enveloped. Enveloped viruses are a little different because they have this biological membrane that surrounds the capsid and the viral genome. The biological membrane is made up of lipids and proteins. Virions are only infectious if the envelope is completely intact. If the envelope is destroyed in any way, then it is no longer infectious. Viruses are called obligate intracellular parasites that infect host cells. Intracellular because they go inside the cell. Once the virus enters the cell, the viral genome is released from the capsid. So, once the virus enters the cell, if the genome has DNA, then it will affect the transcript process of replication. If the genome has RNA, then it will affect the translation process of replication. Alright, so I have this diagram that shows how enveloped viruses enter the host cell. So, the biological membrane is made up of proteins and lipids, which is the same thing that the plasma membrane of the host cell is made up of. So, when the enveloped virus enters the cell, the envelope biological membrane just goes into the plasma membrane, which only leaves the capsid and the viral genome. Once that is inside of the host cell, then the capsid will release the viral genome, and the viral genome will interrupt either translation or transcription, depending on if it has DNA or RNA. And why it does that is because once it affects the replication process, every time that host cell replicates its genome or code, then it will have that viral genome as a part of its code.